as Will said, we're going to talk about furry friends and fossil giants and really think about what we can learn by looking at cancer from a perspective of the history of life on Earth. So not only looking at our recent history, but really going back millions, hundreds of millions of years. So I have some disclosures that are listed here. I won't go through all of them in great detail, but I think most importantly, I just want to point out that all of these relationships are managed by the University of Minnesota in accordance with its conflict of interest policies. So we have a few objectives that I want to achieve for today's presentation. The first is to make cancer less of a mystical disease. So I want to be able to allow you to look at cancer more objectively, particularly thinking about it in the context of how it has come to us in light of evolution. I want to provide background and context for the risks and the causes of cancer, and I think this is really important. I want to explain why cancer appears to be increasingly common and ubiquitous in our society. So really addressing what many people refer to as the cancer epidemic. And I want to describe solutions for this perceived cancer epidemic. Now, at the end of the talk, I hope that you will remember just a couple of really important things. The first is that mutations in DNA, longevity or how long we live, and the effects of natural selection through evolution and the generation of species really account for most of the excess risk of cancer that we see in humans and in companion dogs and cats. And most of the talk will be about dogs. So here's my pointer, dogs. But virtually everything I say, a lot of what I say is also applicable to cats. So for all of you cat lovers, we love cats as well. We just happen to have spent a lot of our time studying dogs. The second thing that I want to leave you with is that emotional changes in behavior are unlikely to alter the risk of cancer in companion dogs and cats. And this means that people are subject to suggestion because of our emotional fear of cancer. And we do a lot of things that really won't make a difference other than emptying our wallets and enriching other people. And then the third point that I want you to be aware of is that work that we're doing at the University of Minnesota and elsewhere looks at how we can address this problem of cancer. And our strategy is to look at risk assessment or understand cancer risk and use targeted prevention in high-risk individuals to reduce the impact of cancer in our society. And this is really exciting work, and I hope that by the end of the talk, you will be as impressed as we are. Okay, so we have a roadmap for what we will discuss today, and there will be 4 small chapters in our presentation. So the first chapter is going to be talking about what is cancer, so that everybody starts from the same page. The second chapter is going to be to discuss whether cancer is a disease of modernity. The third chapter is going to be talking about evolution, how evolution relates to cancer protective mechanisms, and how those cancer protective mechanisms break down when we break the evolutionary barrier on our longevity. And I realize that these are really big words. Hopefully by the end of the presentation, it'll all make sense. And then the fourth one is managing the inevitability of cancer. And this reminds me of when I was in vet school and we had our neuroscience course at the beginning of vet school in the first semester. And our professor said, don't worry, it'll all come together at the end. Well, it hasn't quite come together for me yet, and maybe that's because it's not the end. So maybe my last breath is when I will actually be able to understand neuroscience, but I hope to actually do better than that. Okay, so let's get on this roller coaster ride. So we're going to start today by discussing what is cancer. And cancer is one disease. Cancer actually describes several hundred diseases, and they're all characterized or have in common that they have uncontrolled cellular proliferation. Now, to know that cancer in general, or cancers individually, are all diseases of genes, but they are not heritable, or mostly not heritable. Okay, so unlike other genetic conditions that we think about that are passed from parents to offspring, more than 95% of cancers in humans and virtually all cancers in animals are not passed from parents to offspring. The way that genes are affected is that multiple mutations that alter key cellular functions and a permissive environment, and this is really important, and I'm going to harp on the permissive environment repeatedly, are necessary to initiate and sustain cancer. So many of you have probably heard about driver mutations, and this is what we're talking about here. So the assumption is that somewhere between 4 and 6 mutations can actually make a cell acquire the properties that that cell needs to form a cancer. But there's a lot more that needs to happen in order for that cell to progress onto cancer. So in other words, not every mutant cell will actually give rise to a malignant tumor. The cells need to be able to adapt to their environment. And essentially, it's a microscopic version of Darwinian evolution where the body is trying to prevent the cancer from growing, the cancer is trying to grow, and they're each trying to essentially develop the traits that will beat the other one to see who wins. It's a little bit of that cheetah and gazelle evolution, where the cheetah gets bigger teeth and bigger claws, and the gazelle gets better moves to run away from the cheetah. And so this is a race of evolution. But it's not only cellular evolution, it's also the environment around it. So in the case of the cheetah and the gazelle, they need to have a big area where they can play out their role. And so when we think about cancer, cell division, and mutations, and we're going to talk about this in greater detail later, but I want to take you through this somewhat complicated diagram that I borrowed from my friend and colleague, Dr. James DeGregory. And so James illustrated really nicely that if we think of any generic animal or humans being animals, and we essentially look at age, which is illustrated here on this graph on the x-axis, with 0 being birth, and then infinite being the time when we die. And then we look at the relative scale of each individual pattern that we're looking at from 0 to maximum. Cell division is ongoing at a maximum at birth, and it continues to occur during development and through adolescence. And as we reach adulthood, whatever adulthood means for that particular individual, the rate of cell division, and particularly stem cell division, is reduced. As these cells are dividing rapidly and frequently, we're also increasing the number of mutations, because there's a process that cell division is not perfect. And every time that we replicate DNA, we're going to introduce mutations. And so you can see that mutations increase proportionately or inversely proportionately with cell division, right? And as soon as cell division slows down, we actually slow down the acquisition of mutations, but we acquire mutations throughout life, not only because cells continue to divide, but also because now we are being exposed to things in the environment that can alter our DNA. But basically, throughout development into adolescence and early adulthood, as we have a lot of cell division and as we accumulate a lot of mutations, there's very, very little cancer in the population. And our overall fitness starts at a really high level. So we're born and we really have great fitness in our body and our environment. And that fitness remains at a very high level until a certain point at which that fitness begins to decline. And the decline in fitness is essentially wear and tear, maybe other things that we do to ourselves, behavioral things that we do to ourselves. And so the interesting thing is that we don't really see cancer incidence appear in any meaningful extent in the population until we start seeing that fitness decline, despite the presence of many, many, many cells with many, many mutations in them. So this permissive environment is absolutely essential. So let's go back to mutations. The risk of mutation is intrinsic to all of the life that's based on DNA. So DNA is the blueprint of life on Earth, but it behaves according to the laws of physics and the laws of nature that govern the natural world. In terms of chemistry, there's really nothing special about DNA. It's simply a molecule And really, the only magic about DNA is that its physical and chemical structure allowed it to outcompete every other self-replicating molecule in the early Earth, sort of in that primordial soup, to become the dominant code of life on Earth. It's an interesting thing to think about, that as we start looking for extraterrestrial life in other planets, a lot of that pursuit is based on looking at DNA, but just because DNA won the race on Earth doesn't mean that DNA would have won the race somewhere else. So I think when we think about, for all of you budding astrobiologists, we really need to think about what other self-replicating molecules might have actually won the race in other planets, and what might that look like. But that has nothing to do with furry friends and fossil giants. So we're gonna come back to our original discussion. So DNA has a 4 billion year history of success on Earth, but it actually has some very unique vulnerabilities. And those vulnerabilities come from the fact that it is a reactive chemical. And by being a reactive chemical, it's actually subject to mutations. So how do mutations happen? Mutations can happen in many, many ways. And the simplest type of mutations is where a base, so DNA is composed of 4 bases, A, C, T, and G. They're in a particular order, and there's a change in that order. So in the simplest case, we can have a new base coming in that we call an insertion. We can have a base disappearing that we call a deletion. We can have a base changing, so we call that a substitution. And that changes the code that DNA uses to create proteins. That mutation will actually create an altered protein that can then alter cellular function. It seems simple and innocuous enough, but it can be quite damaging. So we also need to think about cancer risk and cancer cause. So cancer risk is not the same as cancer cause. For example, for people that use tobacco products or that smoke, they increase the risk. But it's not actually the tobacco that's causing the cancer. The tobacco is increasing the rate of mutation. It's creating a permissive environment, and it is really that which causes mutations that is modifying the cancer risk. And it's the mutations in the permissive environment that are causing cancer. So mutations in a permissive environment together are necessary for essentially giving the cell the traits that it needs to become a cancer, and creating the environment that is necessary for those cells to create the disease. But mutations are not bad. They are the basis for biological diversity. So as we said, DNA is a mutable chemical. Without changes in DNA, without mutations, there would be no diversity in the kingdom's life. And I want to drive this home really, really hard to say, without mutations, we would not be here. Mutations are the foundation for how we became who we are. So if there were no mutations, we would be a bunch of single-cell organisms, and the Earth would be a much more boring place. So the environment around us determines when mutations are favorable, and they really drive speciation in the context of evolution. But again, when they happen in somatic cells and they change the code of DNA, sometimes they can be bad, and they can lead to diseases such as cancer. So when we think about cancer-causing mutations, we think about 3 main mechanisms that can lead to these mutations. One is syndromic or heritable cancers, and these account for about 5% of cancers in people. There's about somewhere in the order of 40 syndromic or heritable cancers that have been characterized. Many of you might be familiar with the heritable cancer syndrome that is responsible for breast cancer in women that have the BRCA1 or the BRCA2 mutation. So those are syndromic or heritable cancers. In this case, the mutation exists in the family, and the mutation is passed down from parents to offspring. Again, it is responsible for a very, very small proportion of human cancers. And in the case of dogs, we only know of 2 syndromic cancers, and both of them have essentially been controlled, and they're now just laboratory curiosities. Then we have sporadic cancers. Sporadic cancers are those that are not heritable, and they're due to intrinsic factors. And so when we think about DNA replication, the enzymes that make new DNA are not perfect, and they introduce one error or one mutation for about every million bases that are replicated during cell division. One cell has about 10 to the 9 or a trillion bases that are replicated during cell division. And so if you think about one error for every million bases. That means that every time that a cell divides, it will acquire, on average, about 1,000 mutations. That's a lot of mutations when you only need 4 or 6 to make that cell a potential cancer-causing cell. And then there are, of course, sporadic cancers. Again, they're not heritable, that are due to extrinsic factors. These extrinsic factors include infectious agents, chemical and physical forces in the environment that cause mutations in DNA, and themselves or through some other mechanism, they can also create that permissive environment for cancer that allows a cancer to progress. So this is the distribution of these cancers is nicely illustrated in this figure from a paper that was published in 2017 from a study done by Kristen Tomasetti and Bert Vogelstein at Johns Hopkins University. And this figure is the cause of a lot of anxiety for a lot of people, and there's some controversy, but I think it just illustrates one point really nicely. And that is they look at cancers in humans that are estimated to be due to hereditary causes. So these are essentially syndromic cancers. The ones that are due to environmental causes, essentially tobacco exposure, sun exposure, other similar things. And then the ones that are due to simple replicative or intrinsic errors in DNA. And their estimate is that about 5% of all human cancers are hereditary. Maybe about 1/3 of all human cancers are environmental, but the other more than half to 2/3 actually are replicative. And this really goes counter to conventional wisdom, where we think smoking, sun, food, chemicals are responsible for all cancer. And yet here we're now saying just being alive, essentially, if you're alive for long enough, that's going to be your greatest risk of cancer, with environmental exposures sort of coming in second, and hereditary factors coming in last. So, what is the evidence? How can we actually say this? So we need to go back and think about whether cancer is a disease of modernity. We have only really been modifying our environment for a few thousand years. So did cancer just start after we started modifying the environment and becoming exposed to these things that maybe our ancestors were not exposed to? So I really love this idea because there are a lot of myths surrounding when cancer arose. And by the end of this talk, I hope that you will appreciate that in any multicellular animal that lives long enough, cancer becomes a risk and a reality. So we'll start out with the premise that animals that are single-celled, bacteria, yeast, do not develop cancer. So if you're an organism that only has one cell, you can live forever and cancer won't happen, because cancer is a disease of multicellular organisms. So let me illustrate this. If you're mutations happen in every living being, but if you're yeast, you don't get cancer. What happens when yeast divides out of control? Well, when yeast divides out of control, you get really cool bread or beer. So, yeast cancer is something that we like to enjoy with a good dinner. However, if you're a multicellular animal, your cells are organized into tissues that have specialized functions. And uncontrolled cell division in any of these organs or tissues that have specialized functions creates stress that hinders the function of the whole organism. So if you think of your liver, you kind of need your liver to live. And if you have a liver tumor that alters the function of your liver, that's going to actually not be very good for life. So uncontrolled cell division can then be life-threatening. And this is true for all multicellular animals. And so if you're interested in reading more about cancer and things like sea cucumbers and sharks and parrots, excuse me, there's this wonderful paper from Albuquerque and others published back in 2018. And it's not a particularly exciting paper. It really is more of a catalog that goes across the tree of life and says, yeah, pretty much every organism in the multicellular world gets cancer. So the other thing that we know is that cancer has existed way far back in the animal world, as far back as we can trace. And so this story appeared in the BBC about a year ago, February of 2023. And I'm going to magnify this if I can. Let's see, maybe I need to. Let me see if I can magnify this. It's not working with the pointer. How do I insert the pointer? There we go. Okay. So I wanted to make this big so you could read it. So it says scientists discovered the telltale signs of a range of dinosaur diseases, and they found that they're remarkably similar to those affecting animals alive today. On a wet, stormy day some 77 million years ago, in what is now southeastern Alberta, Canada, a certain horned dinosaur was having a very bad time. The adult Centrosaurus apertus, a medium-sized plant-eating cousin of the larger Triceratops that lived alongside Tyrannosaur, had an advanced malignant bone cancer in its shin. The cancer may have spread elsewhere in its body, and it's thought that it was almost certainly terminal. Okay, so we actually have a lot of evidence that there was cancer in the ancient world. So we're talking about hundreds of millions of years ago from the fossil record. Cancer is an ancient condition. So this is just another picture of a tumor. This tumor comes from a fossil bone of a turtle. It was a stem turtle or a turtle ancestor from the Triassic period, and it's estimated to have lived 200 million years ago. So why bones? Well, bones are better preserved than soft tissue. So when we find tumors in the fossil record, we tend to find them in bones. But the recent treasure trove of fossils, primarily from China, where we can actually see really well-preserved soft tissues, tells us that soft tissue tumors were perhaps as frequent in the ancient world as they are in the modern world when we think about animals outside of humans and domestic animals. So cancer is not a modern physiological defect, but as Hardy et al. Said in this paper, it is a vulnerability that is rooted deep in the evolutionary history of all multicellular animals. But cancer only becomes a thing for us, meaning a disease of concern in the 20th century. So if we go back and we look at dinosaurs and we look at the fossil evidence, the rate of cancer in dinosaurs that we can surmise was about the same as what we see in modern birds outside of the domestic realm. So just a small percent of the population, somewhere between 2 and 10% of the population. And that's what we see across the vertebrate world. So when we look at sort of vertebrate animals in the wild and we say, what is the cancer affliction in a particular population? It's somewhere in the range of 1 to 10%. Okay, so this is the real reason that dinosaurs became extinct. With thanks to my hero Gary Larson, who was one of the best humorists that I've ever come across. But this allows me to dispense with cancer and dinosaurs and move on and start thinking about what about people, what about humans, and how does this apply to dogs and the people who love them? So there's a really fun story from the book called The Emperor of All Maladies by Sid Mukherjee. And in the book, Sid narrates about the reason behind the Persian invasion of Greece in 500 BC. And so Queen Atossa was King Xerxes' wife. And King Xerxes was the ruler of Persia and one of the most powerful men in the world. It turns out that Queen Atossa developed breast cancer. And one of her slaves, who was a Greek slave, had medical training from when he was in Greece. And so the queen turned to this slave who actually treated her. And he cured her breast cancer. He was able to remove her breast cancer. So the queen was very, very grateful for this. And she asked the slave, how can I help you? What can I do for you? And the slave said, well, you can find a way to send me home. So the queen thought about how she could do this and how she could do this. And she said, aha, I have an idea. So when she was in her quarters with King Xerxes, she said, Xerxes, I am very disappointed in you. The Greeks, they have insulted you, they have made fun of you, and you sit here and do nothing. And so she convinced Xerxes that he should go to war against the Greeks. When Xerxes went to war against the Greeks, she was able to place the slave in one of the boats and give her a chance at freedom when they got to Greece. So unfortunately for Xerxes and his armies, it didn't turn out too well for them. But anyway, the War of Quintosa actually was caused by cancer and the fact that she had a slave who cured her. So we know that cancer has been around for, at least 2,500 years. But in fact, much longer. So we have a lot of evidence from mummies and other parts of human history that tell us cancer has been around in the human population ever since humans became humans. So now we have to talk about evolution, or this gives us a nice segue to talk about evolution. Cancer protective mechanisms, which some of you might have heard about, but some of you might not. And the evolutionary longevity barrier that I suspect is going to be a new concept to many of you. So when we think about size and longevity, longevity being how long we live, size and longevity alone do not fully explain the observed cancer risk across the animal kingdom. So going back to the early 1900s, Professor Theodore Boveri, who is the father of cell biology, was working on cell division, and he hypothesized that the more that cells divide, the greater the cancer risk would be for an organism. So this makes sense based on everything I told you. Cells divide, they introduce errors of replication or mutations. These mutations can give rise to cancer. So all of that is well and good. And then came Sir Richard Peto. So Sir Richard Peto, for those of you who don't know or haven't heard of him, is an English statistician, still alive, that contributed to uncovering the association between tobacco use and cancer. So Peto and Dahl really were the key individuals that understood that tobacco use was associated with a massive increase, not only in lung cancer, but a whole variety of cancers. And it really took their skill, their smart, and their guts to actually push through this concept and convince us that smoking was not only not good for us, but was actually really, really bad for us. But among the things that Peto recognized is that if, in fact, having more cells would require more cell division, and cell division increase the risk of cancer, you would expect that the bigger an animal was, or the longer it lived, both of which would lead to having more cells, would increase the risk of cancer. And so we know that if we look across the animal world, there's sort of this relationship that bigger animals, so bigger size, also tend to be longer lived. And yet these big animals that have long lifespan don't seem to get more cancer. So the question is, why don't elephants get cancer? And so Len Nunney, who is at Riverside in California, sort of enunciated Peto's paradox and said, why is it that there's this disconnect between cell number and cancer risk? So that became known as Peto's paradox. So canine cancer makes Peto's paradox even more perplexing. So if we look at body size and lifetime cancer risk, we see that we have really big animals like elephants and whales. And they're very big, so the x-axis is showing their body size. The y-axis is showing lifetime cancer risk. And compared to humans and dogs, they don't develop a lot of cancer. So, there's this terrible disconnect. When we look at dogs specifically, we do know that bigger dogs get more cancer, okay? So on the other hand, when we look at lifespan, we also see that these are very, very long-lived animals. So African elephants are expected to live about 80 years if they're not poached. Bowhead whales, we estimate they live about 200 years. And again, long lifespan, very little cancer. But in dogs, we see this interesting conundrum that little dogs live longer than big dogs. So generally speaking, big animals live longer. Here, little animals live longer. So what's going on? And by the way, this little guy here that we're illustrating is a naked mole rat. It's a tiny little rodent about the size of a mouse, but instead of living 3 months like a regular mouse, it happens to live about 30 years. So the solution to Peto's paradox is the fact that all of these species, and in fact, every species has developed some form of cancer protective mechanisms. Okay, and I realize that you're saying, well, if we have cancer protective mechanisms, how come a third of us are going to die of cancer? We're going to get there. So we have identified many different cancer protective mechanisms across many different species in the animal world. Some of these mechanisms date back to the common ancestors of all animals, and they're widespread across the animal kingdom. And these are the ones that allow us to actually undergo development, right? So if we didn't have these cancer protective mechanisms very early on when our cells are dividing like crazy and undergoing a lot of mutation, we would all essentially be dead. So these ancient cancer protective mechanisms really are present across the animal world, and they allow for development. But then every species has developed their own unique cancer protective adaptations. And these have arisen many times through the course of evolution, essentially with the arisal of every species and independently from each other. And one of the really important things is that these adaptations are influenced or influence the lifespan of the animals in that particular species, the expected lifespan. So cancer protective mechanisms really work only for about the length of time that our species was adapted to live. And I'm going to cover this in great detail. So I'm hoping that by the end of the next 5 or 10 slides, this will become really clear. So the cancer protective mechanisms address different aspects of cancer vulnerability. The adaptations work best during the evolutionarily adapted lifespan of the organism, and they're integral to each species' evolutionary history. And some of the cancer protective mechanisms are preventing mutations. So we already said DNA is a reactive chemical. It's going to undergo mutations. Mutations happen during cell replication. Mutations happen with environmental exposures. And so some of these cancer mechanisms actually are able to prevent those mutations from happening, from propagating, or they fix them. Other protective mechanisms include altering or slowing metabolism. Excuse me. One other mechanism that is responsible for mutations is the fact that our bodies oxidize things, right? So we breathe oxygen, we use oxygen for energy, and in the process of using oxygen, we create oxygen-free radicals. And these oxygen-free radicals are highly reactive chemicals, and they can also lead to mutations. The faster our metabolism, the more free radicals we have. So if you slow down your metabolism, you're going to actually prevent some amount of DNA damage. Okay. Other species like the naked mole rat, or actually various species of naked mole rats, have developed mechanisms to modify the connective tissue environment that supports their system, so the glue that holds their organs together. And by modifying that connective tissue environment, they're able to maintain this sort of youthful fitness longer than others. And there's probably many, many other mechanisms that we haven't even begun to uncover. So cancers become really prevalent when individuals live past the age that nature intended. So when you look across the animal kingdom, cancer is a very rare disease. Again, it accounts for less than 10% of mortality across most animal populations. But in humans, it accounts for up to about 1/3 of all deaths in the developed world. And other than humans, the only other species where cancer proclaims such a large fraction of lives are domestic dogs and domestic cats. There's other really interesting aging problems with cats. So we estimate that in proportion, fewer cats die of cancer than dogs. But certainly they are affected more than the average wild population of animals. So what is it about humans and dogs and cats? What is so special that we have this curse of having this unique vulnerability or fraction of deaths due to cancer? So to really think about that, we have to think about life expectancy. And human life expectancy has doubled in less than 200 years in the Industrial Revolution. So when you look at human life expectancy from, the earliest history of humans, which is not in this graph, all the way to the late 1800s or the early 1900s, x-axis here shows years. And then y-axis shows life expectancy globally and by a world region. And it doesn't really matter which region you want to look at. The point is that once we reach the Industrial Revolution, we rapidly increase human lifespan. And the expected lifespan of someone born in and around 1900 is somewhere in the order of 35 to 40 years. Within 50 years, that lifespan has increased to about 60 years, and at the present time, it's somewhere between 70 and 80 years. So essentially doubled over less than 200 years. And it's interesting to note that this does not really give our genomes enough time to adapt. We were designed to run for 40 years, and now we're running for 80 years. So this is problematic. The amazing achievement of doubling our lifespan was due to things like hygiene, social protections, and medical care. Okay, it's not intrinsic, it's extrinsic of things that we have done as part of our social construct. But the unintended consequence, or the price we pay for this increase in lifespan, is that we increase the risk of what we call aging diseases once we pass this barrier. And cancer is paramount among those aging diseases. So when we think about dogs, we've actually quadrupled the life expectancy of domestic dogs in the developed world since we made them integral family members about 50 years ago. So before 50 years ago, the dog lived outside. It worked. It was really a replaceable commodity, and dogs died of all sorts of reasons. I remember when I started vet school, parvovirus was still a huge cause of mortality in dogs. But we've made changes. We brought dogs into the house. We improved veterinary care. We gave them social protection, better nutrition. And so now, again, dogs used to live 4 years, and now they live 12. So, that's again, a remarkable achievement. And again, this is a really short period of time. So the dogs have not had a chance for their genomes to adapt to this change. So one of the unintended consequences of dogs aging beyond their original life expectancy, or their evolutionarily adapted life expectancy, is that they also are susceptible to what we call diseases of aging, cancer being paramount. So let me show you this in a different graphical way. The first graph in this illustration shows what I already showed you before. So we're looking at time on the y-axis. So this is years going from about 1700 to the year 2000, or a little bit after. And then we have life expectancy, and the y-axis starts at 30. So the life expectancy before the year 1870 or so is somewhere on the order of 30 to 35 years, maybe 40. And we rapidly increase life expectancy in the years thereafter through all of our social constructs. When we look at the deaths corrected for 100,000 people and the age range, we see that in our evolutionary adapted lifespan, which is this dotted line, there's very little cancer. The proportion of deaths per 100,000 people due to cancer is almost imperceptible. But once you reach the ages of 45, 50, 60, 70, and so on, the deaths attributable to cancer increase dramatically. So why do we not see a lot of cancer in the ancient world? Well, mostly because people were just not living long enough to get cancer. And I'm going to get to this illustration about lung cancer in another slide, so I'm going to skip it for now. And what I want to show you here is a comparison of what happens when we actually surpass that evolutionarily determined lifespan barrier. So what we have here, again, on the x-axis of this graph on the left is lifespan. And on the y-axis, we have the species that we've talked about before. And they're not the only species that have cancer protective mechanisms, but they are ones that illustrate the point really nicely. So we have bowhead whales, African elephants, naked mole rats, and then we have ancestral humans and ancestral dogs. So the orange colored bars represent species that have a lifespan based on evolutionary time, or the lifespan that they originally adapted through evolution to live. And so over millions of years, bowhead whales adapted to live about 200 years. They actually acquired a lot of genetic traits that gave them the opportunity to invest the energy to have long gestation periods, babies that needed to be nurtured for a really long time, with an expectation that they and their babies would live a really long time. African elephants evolved over millions of years, so about 40 million years of evolution in the elephant lineage, to live about 80 years. Naked mole rats evolved over about 40 million years, side by side with other rodents. The difference was that these guys actually evolved to live about 30 or 40 years. And they can do that for some really simple reasons. They live underground. There are many fewer predators that eat these guys. And so they can therefore invest energy to have long, productive lives in whatever a naked mole rat does. And then we have ancestral humans. So to the best of our ability to understand the lifespan of ancestral humans based on history records, based on modern nomadic or undeveloped societies, we can estimate that ancestral humans evolved to live about 40 years. And we know for the same reasons that dogs evolved to live about 3 to 4 years. But modern humans, so now modern outlined in blue, live 70 to 80 years, and modern dogs live 10 years or longer. Or at least that's the expectation. And so when we look at cancer incidence in humans, now we have human age in years from 0 to 100. We see that there's very little cancer over this evolutionarily adapted lifespan. And as we cross that lifespan, we see a rapid increase in cancer occurrence and cancer deaths. And the same is true for dogs. So when we look at dogs, so this is dog age in years. So this is not converted to human. This is the actual dog years as we perceive them. Again, the evolutionary adapted lifespan of dogs is somewhere on the order of 3 to 4 years. Very, very little cancer incidence or mortality during that time, rapidly increasing as dogs reach an age that goes beyond that evolutionary adaptation. So we have to think about natural selection and how natural selection drives adaptations to a particular niche. So species adapt to these environmental niches over millions of years of time. And the adaptive traits that protect from cancer allow species to occupy niches where they can invest energy to become large, have a long lifespan, or both. And again, these changes are stochastic, they're random, they're not directed, and they occur over millions and millions of years. And they are driven by the same processes that drive evolution, which is not specifically just like, are you good at reproducing? There's a lot of other things that address macroevolution. So as I said, there are many, many known cancer protective mechanisms across the animal world. And if we sort of match the mechanisms to the species, many species of whales and other aquatic vertebrates like fish, sharks, for example, are able to slow their metabolism. And so they create less free radicals. They limit the damage to their DNA. And there's actually other protective mechanisms that we've been able to elucidate from whales. Elephants and others have repeated duplication of a tumor suppressor gene called P53. There's some really interesting data on what these new copies or different copies of P53 might do or can do. And there's evidence that there's other genes that have been modified or duplicated over evolution that promote cancer protective abilities, or that have cancer protective abilities. We talked about naked mole rats and how they alter their connective tissue homeostasis to prevent cancers from being able to effectively grow. In humans, we don't really know what our specific cancer protective mechanisms are. We know that there's a mechanism that we call telomere attrition. As cells get old, the ends or the tips of the chromosomes start shrinking, and that prevents the cells from dividing when they reach a certain age or a certain number of cell divisions. That's shared with many species. We obviously have others that we have not encountered. And the same is true for dogs. Despite a lot of studies, we don't know what all the mechanisms that protect dogs from cancer through their evolutionary adapted lifespan are. We know that there is telomere attrition, which is shared with other species, but there's other mechanisms that we haven't found yet. So we're going to go back to this idea of the permissive environment. A consequence of outperforming our evolutionarily determined life expectancy, which would end here, is increased cancer risk. Okay. Now, some people might say, well, that's all well and good, but we know that things like smoking cause cancer. So now I'm going to actually show you what happens when you put this model in the context of the most powerful group of mutagens that we know. Okay. And that's cancer. So these are data from the British Health Service. And what they've done here is they've taken patients that smoke And then they've looked at the incidence of cancer in men in blue and in women in pink. And so one of the things that you have to think about is we have age at diagnosis, right? And so it's an increment. So this is 0 to 4, 5 to 9, and so on. Virtually every smoker starts smoking somewhere in the preteen to teen years, okay? So this is where people start smoking. And they will smoke for the better part of 30 years without developing any signs of cancer. All of the time, there's damage happening, but there's no overt cancer, okay? And once they reach the age where there is a permissive environment that's being formed through aging or outliving our cancer protective mechanisms, boom, we start seeing this increase in cancer. So the hypothesis is that if in fact it were purely tobacco, you would see cancer starting here. And there's been many hypotheses proposed for why we see this lag time with tobacco and other compounds. But I think this explanation is really as satisfying as any. Now, fortunately or unfortunately, as the case might be, it would be unethical for us to do the experiment to actually prove this, where we would take non-smokers at the age of 45 to 50, get them to smoke 3 packs a day and see if they develop cancer within 6 months. That's what this model would predict, but of course that is not an ethical experiment, so it will never be done. So I think the point that I'm trying to drive to is that we really need to be open-minded about how we think of cancer risk, cancer exposure, cancer costs, and not necessarily attribute it to what we might think is the most obvious thing. So tobacco, mutations, cancer, but how do we explain the time difference, right? So if we think about cancer in this context of aging and cancer protective mechanism, Peto's paradox actually becomes Peto's principle. And that is because evolutionary natural selection increases the probability that beneficial traits, including cancer protective mechanisms, will be passed on to future generations. So cancer protective mechanisms allow for successful competition in niches that accommodate these animals with large body mass and long lifespan. Okay, so their adaptive changes or their whole adaptation to that environment through evolution is what allows them to be big and long-lived. And so Peto's paradox really is a result of superimposing adaptive evolution driven by natural selection on populations. And so when we look at elephants, they're really big, they have a lot of cells, they live a long time. Why don't they get cancer? Because evolution actually helped them be able to achieve that size and lifespan. So when we look at domestic dogs, there is a difference now because we have now taken out or modified natural selection and replaced it with artificial selection. And we have taken advantage of the plastic nature of dog genomes and made them huge and tiny. And in fact, what we see is that longevity and cancer risk in dogs, as shown here, life expectancy and the y-axis, decreasing, increasing as you get smaller, cancer risk generally, osteosarcoma risk in particular, increasing with increasing size. And so now again, dogs are showing us that Peto's paradox is not a paradox at all, it's a principle. I know we're at 40 plus minutes. I'm gonna go another couple of minutes and we did add some times for questions. So I wanna say that the effects of size and aging on canine cancer risk are so strong that they pretty much render other causes insignificant. So what are those possible causes? Breed, nutrition, chemical and environmental exposures, spaying and neutering. So yes, there might be breed influence, but compared to size and longevity, it's pretty minor. And in fact, there's 2 papers that just came out in the last week, one from the Dog Aging Project and one from Len Nunny that actually shows that when you look across the whole of dogdom and you consider size, most of the breed differences in terms of cancer risk go away. Not all, but most. When we think about nutrition, there's a beautiful study from the Purina Lifetime study done about a decade or a little bit more than a decade ago that shows that it's not really how you eat, it's how much you eat. So reasonable dietary restriction not only extends life and improves the quality of life, but it's also cancer protective. So it's not whether or not you have enough vitamin this or enough, fatty acid that. It really is sort of the overall balance of your diet and how much you eat. Overnutrition is not good. When we think about chemical and environmental exposures, there are many, many studies looking at chemical and environmental exposures. And the problem with these studies is that they're generally not reproducible. So there's a brand new study that came out also a couple weeks ago from Purdue showing an increased risk of cancer in dogs that are exposed to environmental tobacco smoke. I don't doubt that there's probably some effect. However, when people have looked at this question repeatedly, they don't always come up with the same answer, and oftentimes they come up with a different answer. So we do know that if you have sort of laboratory exposures that are really above and beyond what any normal animal would get, you can create absolute, horrible cancer risks. So if you take dogs and you expose them to levels of radiation that would be only caused in disastrous reactor meltdown, you will actually see increase in cancer. But when that happened as an experiment of nature in Chernobyl, we actually have seen that there's no increase in the relative rate of cancer in those dogs. So we can cause a lot of cancer with radiation, but in fact, the chemical radiation meltdown, which we expected would lead to terrible increases in cancer, did not. And then spaying and neutering. Spaying and neutering is sort of the bane of my existence. There are reasons to spay and neuter dogs. There are reasons not to spay and neuter dogs, but cancer is not one of them. So what we know is that hormones are associated with mammary cancer in dogs. Keeping dogs intact over several heat cycles increase the risk of females to develop mammary cancer later in life. And that's mostly due to hormones, estrogen. When we actually spay and neuter dogs, one of the things that we notice is that it increases their lifetime or their lifespan. And why is that? Well, probably because many owners that spay and neuter, doesn't mean that owners that don't are bad, but many owners that spay and neuter are taking better care of their dogs. They're giving them veterinary care, they're giving them shelter. And so when you actually look at the excess cancers that have been reported in spayed and neutered dogs, virtually all of that excess cancer is explained by the fact that the dogs are living longer. And so they get more cancer because they're around for a longer period of time. And then my favorite story from vet school, Russian lasers. So we had a client when I was in vet school that brought her little dog wrapped in foil. And she wrapped her little dog in foil because she was sure that Russian spies were shooting lasers through her apartment window trying to give her little dog cancer. And so she wrapped her little dog in foil to prevent the Russian lasers from giving it cancer. I can assure you that Russian lasers are not the cause of our cancer epidemic. They're probably the cause of a lot of other ills, but not our cancer epidemic. Okay, so how can we actually tell that aging is the cause of most cancers? How can I back up what I'm telling you? And so we now are able to look not only at the mutations, but at the signatures that they create around them. So when we look not only at the mutant base, but we look at everything that's around it, we can see some patterns, okay? And there is what's called a cosmic catalog of mutational signatures, and many of these signatures are related to very specific exposures or things. And when we look at dog cancers that have undergone this analysis, about 60% of dog cancers, of all the types that have been looked at, are associated with cellular aging and errors of DNA replication and repair. 40% are associated with things that we don't know what causes them, but it's not clear environmental things like tobacco, aflatoxin, other things that we know about, sun. And the ones that are caused by known environmental carcinogens, like I said about, is virtually zero. So again, we have now evidence from the cancers themselves that are telling us it was not some environmental exposure. It was really what's happening intrinsically in the cells. And going back to this sort of interesting difference between dogs and the rest of the animal world, we know that in dogs, bigger dogs live a shorter period of time. And that we think is because as we increase dog size beyond what dogs evolved to do, we create organismal stress that accelerates aging. So in summary, dogs conform to Peto's prediction, and that's because 20,000 years of artificial selection with emphasis on size and 50 years of living indoors have not allowed these large and giant breed dogs to develop cancer protective mechanisms and compensate for this increase in size. And the genomic plasticity that achieves variations in phenotype has not been exploited to the same extent in other animals. So in the last couple of minutes, I want to tell you the really cool thing about how we're managing this inevitability of cancer. So if cancer is inevitable, what is the solution? Well, one solution is to actually look at risk in individuals by identifying the earliest changes in cellular transformation, so looking for these mutations, or changes in that fitness environment that create the tumor niche. And I'll tell you that there's many methods that are being currently developed and deployed. But I want to ask you, what if I told you that we have a test that detects cancer risk in dogs 4 years or longer before tumors ever develop? Not only that, what if I told you that we have a solution that modifies this risk by changing the environment that permits the cancer to form and thus delaying or preventing cancer? I think that you would probably say, really? And this is the nicest thing that I could think of that you might say to me. You got to be kidding. Some of you might use stronger language. So let me show you what the test does. So this is what we call the SOS test. SOS stands for Shine On Suspicion. And what we've done here is we've taken 200 dogs, run them through our test, and we can divide the dogs into predictions where we actually have an algorithm that says that there's a particular pattern looking at a very simple blood, well, that's not a simple blood test, looking at a test that we use by taking a little bit of blood and then running it through some really cool testing and artificial intelligence algorithms. And we can divide dogs into low risk, which are the orange line, or high risk, which is the blue line. And so time zero is when we run the test. The x-axis is telling us the rate at which dogs develop a malignant cancer, malignant disease. And what you can see is that the dogs that are at low risk develop virtually no disease for the first year, and maybe a little bit of disease through the second year. And this is a single test. And remember, time is ticking, right? So these dogs continue to increase their risk over time. However, the dogs that test high risk out in the beginning, we start actually detecting cancers that were probably already there quite early on. And the increase in cancer rates continues. So what happens when we look at these dogs about 6 months out? Well, it turns out that we see this difference. The dogs at low risk have virtually no cancer. The dogs at high risk, about 10% of them have developed cancer. What about at 1 year? Well, by 1 year, we have maybe about 3% of the dogs in the low risk category that develop cancer, and we now have about 22% of the dogs in the high risk category that develop cancer. What about at 2 years? Well, by 2 years, we would have wanted to retest these guys because it would bifurcate again. But even without bifurcation, we have less than 10% of these dogs in the low risk group that have developed cancer, and now we have about 30% of the dogs in the high risk group. And if we follow these guys out to 4 years, the high risk group will actually get down to about 50% of the dogs in the high risk group will develop cancer. And remember, this is a risk profile. It is not necessarily an absolute yes or no. Okay, so if we tell you, well, your dog has 50% chance of developing cancer over the next 4 years, but there's nothing we can do about it, that's totally uncool, right? It just creates stress. It changes your relationship with your dog. So what happens? Well, we have a solution. We call it an interception, and I'm going to illustrate this interception using a single case. So this is a dog that was tested over here and tested with high risk. And at this point, the dog was about 9 years old, if I remember correctly, 10 years old. We retested the dog several weeks down the road. And what we found was that she was still high risk. So we did a third test, and she was still high risk. So at this point, we offered the owners that they could come for the interception. And what we do is we bring the dog in, we screen the dog to make sure there's no obvious cancer. If there's cancer, they become a cancer patient. They go over to treatment, but if there's no cancer that we can detect, they can go to prevention. So before prevention, the dog is still high risk. Immediately after prevention, the dog is still high risk. But then we go out 4 months and the dog has changed from a high risk individual to a low risk individual. And what happened to this dog? This dog lived another 1,327 days, or 3.63 years from its first test, or 1,185 days, which is 3.25 years from EBAT. And unfortunately, she did die of fragility when she was 13 plus years old. So when she died, she was a 13-and-a-half-year-old boxer with no evidence of cancer. So this is ongoing work. We've treated 8 dogs with our interception strategy. The side effects are really mild and transient. We believe that the interception is safe enough to use in healthy dogs. The treatment or the interception is really well tolerated. 6 dogs have been alive with no evidence of malignancy almost 2 years. The other 2 dogs have died of causes unrelated to cancer. One at 10 months died of a heart condition. The other one that I just told you about died because time goes on. And the other 2 dogs have only been recently enrolled, and so we're just following them along, and they're doing great. And there's this really great story of this 11-year-old golden retriever. I'm sorry that it says you met her here. You didn't meet her here, actually. This was for another event. But she received our interception about 2 years ago, and the family reports that from the time she received this, she has been rejuvenated. So she went from being sort of like an older couch potato to this crazy dog that runs in the pool, swims in the ocean, steals dog from the table, and does all sorts of naughty things that the owners love because she's now acting like a young dog. So again, it's early data, but we're very excited about it. And I do want to mention that our strategy is very consistent with the recommendations that were just released by the Minnesota Governor's Task Force on Academic Health. So they recommend that we invest more in prevention strategies that could have the most impact on the health of the population, that we tap Minnesota's unrealized potential to develop innovative models of prevention and care, and that we align our goals and our expectations with the principles of equity, work for diversity, geographic accessibility, and primary care and prevention. And we are doing all of those. So to conclude, cancer is an ancient disease. It's been with us for hundreds of millions of years. The excess cancers that we see in companion animals are an unintended consequence of our success in increasing their lifespan. Cancer is a very emotional condition, and it's an easy one for predators to exploit. And science can generate objective solutions to our most pressing problems. So I want to leave you with a reminder of our take-home message. So mutations in DNA, longevity, and natural selection account for most of the excess risk of cancer that's seen in humans and in companion dogs and cats. Emotional changes in behavior are unlikely to alter the risk of cancer in companion dogs and cats, and risk assessment and targeted prevention provide one novel strategy to reduce the impact of cancer in our society. I want to very briefly just show you a list of our collaborators, both in my lab and within the U of M and outside collaborators. This is not a story of me. It's a story of a very large team with a lot of support from wonderful foundations, the federal government, your tax dollars, as well as the dogs themselves who have taught us a lot. So with that, I just want to remind you that among many other things, philanthropy drives our work. And you can join us in the fight against cancer by contributing to research that serves animals and people. You can go to our website if you're interested in contributing, or if you want to learn more about impact opportunities that you can do with the University of Minnesota College of Veterinary Medicine, please contact Jen Scholl. Here's her email, and here's her telephone number. And as Will said in the beginning, this will be recorded, so you can go back to this slide and make those notes. So with that, I want to show you my fur babies for whom I get up every morning, and happy to take some questions. Thank you.
Thank you, Jaime. That was a great presentation. Appreciate how thorough that was. We do have some pre-submitted questions, and I'll get to them in the order they were received. So we have a question here about prevention. This person wonders what medical tests or diagnostic procedures should they be concerned with or inquire about as part of a pet's routine physical?
Yeah, that's a really good question. So, we are living in an era of tremendous innovation and transition, and there's a lot of stuff out there. And the question is, what of this stuff do we use? But the better question is, what of this stuff do we know how to use? So I think that um, having a routine physical exam by your veterinarian with follow-up routine laboratory work like blood tests, complete blood counts, serum biochemistry profiles, urinalysis, allow your veterinarian to assess whether there are changes happening over the life of your dog that need attention. And so the routine tests are really sort of the baseline recommendation. What else should you do? And the real answer, the most honest answer, is we don't know. So should you be doing imaging to catch cancers early? There are people who do that, and it's not contraindicated. What we have seen is that there are dogs that for which we have found tumors early, and we can go ahead and treat those tumors. Sometimes those dogs do better than we would have anticipated, and sometimes they don't. So there are data that tell us that generally tumors are born to do what they're going to do, and so early detection may not always work because a bad tumor is born bad, and a good tumor is born good. And so sometimes going after good tumors that are not going to do a lot of damage increases morbidity. So there's that level of uncertainty. We have seen people that image their dogs, and the dogs go through an ultrasound or radiographs, and they come out clear, and 3 weeks later they have a giant tumor. So sometimes these tumors can grow very, very fast enough that we don't see them even with imaging, and then we see them very shortly thereafter. There are newer tests that use sort of the power of genetics and molecular biology. And again, the biggest problem is that we don't have enough data to determine what we can do about those tests, and they are not always actionable. So if you run a test and it tells you that your dog might have a cancer present, how accurate is the test, and what do you do about it, right? Maybe you go and you do imaging. If you find the tumor, you're back to, is it a good tumor? Is it a bad tumor? What do we do about it? If you don't find it, but you have in the back of your mind that your dog has cancer, that changes your relationship with your dog. In an extreme case, that change in relationship might be so stressful that you choose to put your dog down. In other cases, it may be that you choose not to put your dog down, but your dog has an accident and you decide it's not worth treating my dog because it's going to die of cancer. So we really are thinking about not only what tests are available, but what can you really do about them? And the answers are not straightforward. So what do we recommend? We recommend to stay with a conservative routine testing. If you want to sort of be an early adopter and get ahead of the curve, just recognize that you are adopting something for which we may not have really good answers.
Thank you, Jaime. We have a question about treatment. This person asks, what are important things to do in order to support a pet as it's going through chemotherapy? Anything for pet owners to keep in mind?
Yeah, that's a really good question. So transparency and disclosure, I am not an oncologist. I am a pathologist. So I do not treat dogs myself. And so let me answer the question based on the best understanding that I have from working with colleagues who are oncologists. In veterinary medicine in general, and in veterinary oncology in particular, the goal is to provide the best quality of life with the most life extension we can achieve. So generally, as a profession, we don't go for life at any cost. We go for highest quality of life for as long as we can. So we want dogs with cancer to behave like dogs without cancer. We want to actually give them comfort and life extension, recognizing that we won't go that extra mile that could achieve a potential cure, but is going to actually put them in ICU for 3 months. So I'm very proud to say that dogs in whose care I have been involved either as a pathologist or as a researcher, but have been treated by my colleagues, have gone on to win agility championships perform exceptionally well at dog shows, do free ball and all sorts of really cool dog activities, become master hunters during the time that they were getting chemotherapy. So the question is, what did the owners do to support that? One is they worked very, very closely with the veterinary care team to understand what side effects might be coming, how you manage them, how you anticipate them. Nutrition is important. You want to give the dogs the balanced nutrition. You don't want to overnourish them, and supplements, in one word, are pretty worthless. I guess that was 3 words. So I would invest time and energy into giving your dogs mental activity, physical activity, a lot of love. If you really want to understand the best nutrition, talk to a board-certified veterinary nutritionist. Most of the time, you can achieve that balanced diet with commercial diets. You can achieve the sort of love with commercially available or homemade treats. Do keep your dogs lean. That makes a difference. So I think that the short answer is common sense.
Thank you, Jaime. We have time for one last question. And this is a question from someone who asks, for people involved in dog breeding, are there any behaviors or tests that breeders should be considerate of when in the course of their work?
I'm not sure that I totally understand the question. Let me see if I can rephrase the question that I will answer. Should breeders look at available tests in making breeding decisions? I think that generally the answer is yes. Should breeders think about cancer risk or cancer in the line as part of their breeding decisions? The answer is generally no. So we only think of heritable cancers in the context of those cancers affecting not sort of a population of old dogs at large, but really resulting in multiple cancers within littermates or siblings at a very young age, where those cancers have something in common. So in dogs, a young age would be probably somewhere under 4, and it would affect a vast proportion of the dogs in a litter. So it wouldn't be 1 or 2 in a litter of 10. It probably would be 6 or 8 or 9 in a litter of 10. And those cancers would occur at a young age. Most of the time, there would be more than one cancer, and they would have something in common. So it is really only under those circumstances that we start thinking that there's a heritable condition. And for people that are interested, looking at the pedigrees and the ages of the dogs in the syndromic cancers that have been published. So one is called RCND in German Shepherds, renal cystadenoma and nodular dermatofibrosis. I always have to think about what RCND stands for. Beautiful set of papers from a group in Norway with collaborations across the world. So when you look at those pedigrees, you'll get an idea of what a heritable cancer syndrome looks like. And then a single paper published by a Japanese group in Parsons Russell Terriers or Jack Russell Terriers with a mutation of the APC gene that developed colon cancer, very much like human families that have a mutation in the APC gene. And again, those pedigrees, the sheer number of dogs that develop it and the age is really striking. So I think that if you think about, I have a dog and its dam developed cancer and now should I breed it, or its sibling developed cancer and now should I breed it? That really should not play a role, because we're really talking about all these other factors and they're not necessarily embedded in that dog's genome.